We're going to cover Chapter 19, and we're going to talk about epidemiology. So remember, at the beginning of the semester, we talked about how Semmelweis looked at how women were dying after childbirth of puerperal fever. It's actually a bacterial infection of the uterus, and he realized that the medical students were working with cadavers and not washing their hands before they would help a woman deliver, whereas the midwives were washing their hands and they had lower rates of death. He actually forced the medical students to start washing their hands before doing surgeries and ended up having a significant decrease in the number of deaths after birth because you weren't getting these bacterial infections. So when you're talking about epidemiology, it is the study of distributions and the cause of disease in populations. Basically, epidemiologists will collect and compile data about sources of disease and different risk factors. They will design infection control strategies, prevent and predict the spread of disease. They can have expertise in a number of different disciplines, including ecology, microbiology, sociology, statistics, and psychology. A lot of our daily habits that we take for granted, like removing of waste, washing our hands, using a different cutting board for chicken versus vegetables, are all based on epidemiological studies. So when you look at epidemiologists, they have different terms that they apply to the infectious disease process. You can have communicable and non-communicable diseases. Communicable diseases are contagious. They are transmitted directly from one host to another, something like the flu or a cold or the measles. Transmission is determined by interactions between the environment, the pathogen, and the host. You try to actually control these factors to break the infection cycle. So, by washing your hands, maybe you prevent the spread. You vaccinate to increase host resistance. With non-communicable diseases, you do not have spread from host to host. These are going to be microorganisms a lot of the time that come from the patient's own microbiota, and something throws off homeostasis to allow them to become opportunistic. Now, with epidemiologists, they're more concerned about rate than absolute number of cases, right? So it doesn't matter if you have a small city or a large city, they want to know the percentage that are infected, not just the raw number of cases. So the attack rate is the percentage of people who become ill in the population after exposure. This can actually reflect the infectious dose as well as the immune status of the population. The incidence rate is going to be the number of new cases over time over the population size. This is actually a measure of risk that the individual has of contracting the disease. Prevalence will be the total number of cases at any time or for a specific period in a given population. This tends to reflect overall impact of disease on society. This can include old and new cases as well as the duration of the disease. These tend to be expressed as cases per 100,000 people. Now, morbidity is the incidence of disease in a defined population. When you look at contagious diseases, they often have high morbidity rates. Someone who's infected can transmit it to several other people. Now, mortality is the overall death rate in a population. And the case fatality rate is the percentage of the population that dies from a specific disease. One of the reasons that Ebola freaks people out is because it has a very high case fatality rate. Now, when you talk about endemic diseases, these are things that are constantly present in a population. When you have sporadic diseases, you only have a few cases from time to time. An epidemic is when you have an unusually large number of cases. This can be from an introduced disease or something that's endemic. An outbreak is a group of cases at a specific time dealing with a specific population in a specific location, and a pandemic is a global epidemic. Now, when you're studying disease, you have to figure out the chain of the infection. What is the reservoir for the infectious agent? How does it exit that reservoir? How is it transmitted? How does it enter the host? And is the host susceptible to the infection? So with reservoirs of infection, this is the natural habitat in which the pathogen lives. It can be in or on an animal, in or on a human, or in the environment. It can live in the soil and the water. Identifying the natural habitat can be very important in disease control. For example, we know that rodents are a reservoir of plague. So you actually try to control rodent populations in the Midwest to prevent plague epidemics. Now, with human reservoirs, you're dealing with things that are found only in humans or exist in other animals. And really, if something only is found in humans, it's much easier to control. So the reason we were able to eradicate smallpox is because it was only found in humans. The issue here, though, is that individuals who are infected can be symptomatic or asymptomatic. So with symptomatic infections, there's an obvious source of the pathogen. With asymptomatic infections, these are harder to identify because the carriers might not realize that they're infected and they spread it to others. An example is up to 50% of women infected with gonorrhea do not have symptoms, so they're transmitting it to their sexual partners not knowing they have it. A lot of people also carry Staph aureus. We can have non-human animal reservoirs. These can be very common for certain gastrointestinal diseases and rabies. And with zoonoses, these are zoonotic diseases that are found mostly in animals, but they can be transmitted to humans, and unfortunately they have more severe consequences in humans than in the animals in which they're found. You can also have environmental reservoirs. Pathogens found in the environment itself really will never be able to get rid of. So for example, Clostridium and Bacillus are both soil bacteria. We're never going to get rid of dirt. You also want to look for portals of exit and portals of entry. So basically it's a body surface or orifice. It's how the pathogen enters and exits the body. So the intestinal tract, you shed things through your feces, things can enter through your mouth. Same thing with the respiratory tract. You can breathe things in, but you can cough things out. We're constantly shedding skin. And remember, to cause disease, not only must the organism be transmitted, it has to be able to grow. Now, in terms of disease transmission, we can have vertical transmission and horizontal transmission. Vertical disease transmission is when a pregnant woman passes it to the fetus, or the mother passes it to her child during childbirth or through breastfeeding. Horizontal transmission is person to person, and it can happen via the air, physical contact, ingestion, or through a vector. So when we're talking about direct contact, we're talking about something like a handshake or sex, right? You're touching another person. And you can also have fecal-oral transmission falling into this category because you touch somebody else and then they touch their mouth with their hand. Hand washing is actually the single most important measure for preventing the spread of infectious disease, especially things that are spread through direct contact. Also, some pathogens can only be spread sexually because they require intimate contact. They cannot actually survive in the outside environment. With droplet transmission, we're talking about respiratory droplets. These fall about 3 feet away from your mouth, but when you're dealing with densely populated areas like schools, colleges, military barracks, these can be an issue because there's overcrowding. And you can minimize the spread by covering your mouth when you sneeze or cough. You can have indirect contact. So a lot of respiratory diseases are transmitted through the air. So larger particles get trapped by mucus, but smaller particles can enter the lungs and have pathogens. When you laugh, sneeze, talk, sing, cough, you're actually generating particles that are leaving your mouth. And what happens is droplet nuclei form. Basically, microbes attach to material you're coughing up, and they can remain suspended in the air because they're so lightweight. Typically, the number of bacteria in the air is proportional to the number of people in the area. This can be very difficult to control, and this is why you need ventilation systems, negative pressure, and HEPA filters in certain places. You can also have vehicle-borne transmission. So you can have fomites. Fomites are inanimate objects. It's clothing, it's the desk, it's a chair, it's a drinking glass. You can have food and water that become contaminated. So a lot of animal products come in contact with animal poop. So you have to be careful of cross-contamination. Also, if you have contamination of municipal water systems, that can get distributed to a lot of people very quickly. We also have vector-borne transmission. So vectors are going to be living organisms that carry a pathogen. The most common vectors are the arthropods, which is why we talked about them in chapter 13. Now, they can either transmit things mechanically or biologically. So mechanically means they touch something and then they touch something else, so they move the bacteria from one place to another that way. Biological means they actually will bite you, get infected themselves, and then bite somebody else to infect that new person. Typically with diseases that are transmitted via vectors, controlling the vector is very important in preventing the disease.